here at Youngstown State University. I'm majoring in exercise science, and I plan to pursue my PhD in physical therapy. And this is sort of like the first step in that. I'm sort of going to go through this introduction point like a little quick because there's a lot to cover. Basically, this is just an overview of my honors capstone, and I have it linked at the end. But I'm going to talk about the Athletic Truth Group, who they are, exercise physiology, how that plays into this, the Garage Gang, distinctions between two different theories of muscle contraction, and how it applies to mechanobiology. So first, the Athletic Truth Group. So this is where I interned for my spring semester here for exercise science. It's an online training service that utilizes novel exercises from the group's founder, Ben Patrick. And they tout a bulletproofing effect for every joint of the body with their approach. And they have found nearly 2,000 unasked for success stories from people who have gone through the system. And I myself, as I was interning, I probably pulled up maybe 50 or 100 through my time with them. They have a variety of different reasons they think their system works, and the main one I'm going to touch on at the end is strength curves, but I'm going to continue for time's sake. So my reverse research journey, so to speak, actually began in fall of 2019 as I started to look into the system for benefits in athleticism and durability and things like that. But in the spring of 2020, my other mentor, Dr. Basso, he actually presented a lecture on muscle contraction and briefly mentioned the Titan theory. And I'm like, I thought like muscle contraction was done. We already got all the theories, you know what I mean? So it was new. So it really intrigued me. So I looked into it a little bit more. And after trying to connect the pieces with this program and the theory, I'm like, oh, whoa, I think there's something here. So I started training myself 100% compliant with it. I wasn't like, just dipping into it. I actually went full on into it. And I thought it to be safe enough to train my brothers and my friends last summer. And I'm just going to skim through the results. If we have time, I'll go back to just, look into them more deeply. But there's progression results for myself, people that I've trained through this system, and so on and so forth. And what I really want to hone in on, that's actually one of my online clients, which is really, really cool to see his progression. But right here, this is my exercise science professor, Steve Donahue. And long story short, we enjoyed this programming so much and liked what we were finding from it that we're actually partnering up with a business venture to become ATG certified. They're actually certifying people in this system. So that's like a little background. Now this is what I want to really zero in on, is why I think this system works. It's going to be a distinction between two theories, the sliding filament theory of muscle contraction and the Titan filament theory. So before even getting into that, just a brief overview of muscles, tendons, and things like that and how they work. So basically, you got muscles, which connect the tendons connect the bone, which initiate movement at the skeleton. And encapsulating all of these things is a thing called extracellular matrix. It's a connective tissue encapsulation that envelops all the muscle fibers, the tendon, so on and so forth, and it provides a mode for communication, transmission of force, and things like that between all of these different pieces. This arrow is pointing to the myofibril, which holds a bunch of different sarcomeres, which are the functional unit of muscle contraction. That's where it initiates at. You got the I band, and the A band, which refer to actin and myosin. And I think this little thing's supposed to, yeah. So it actually, myosin fibers pull actin fibers closer towards the M line, which shortens the H zone. And that's just a fancy way to say contraction. So it contracts at the muscle cell and goes all the way up the chain, shortens the muscle, pulls on the tendon, creates movement at the skeleton. So that's basically where this theory is grounded on. But I'm going to now go into some research on Walter Herzog on what he thinks are problems with this, possible solutions with this, and where to go forward with this. So the big point of his research, and all of these are cited at the end with my paper. There's way too many citations to do it throughout this. But the big thing I want to touch on right now is that the creator of that sliding filament theory told Walter Herzog he never wanted it to be published. He thought it to be too preliminary and needing more refinement. But this is the foundation of what we know about muscle contraction, which I thought was very, very interesting going into it. So some other interesting facts as we look into this theory. Number one, it doesn't account for residual force enhancement. And what does that mean? Basically, as a muscle actively lengthens, so it's resisting contraction, but it's still actively contracting, it develops more force at a certain contraction than if you just pulled a muscle to a certain length and then contracted it. And that could be seen here. So from zero, my mouse, yeah, from 0 to 10 here, millimeters, you can see that there's more force developed from that point at the black line at any point along that length versus, say, that orange one, which might be at like 6.2 going to 10 there. With the sliding filament theory, the only way to develop force is by cross bridges, and that doesn't account for this history-dependent property of contraction. The next point, instability. This theory predicts instability at long sarcomere lengths, which is one of the reasons people shy away from full range of motion training. But I want to talk about a little bit about this graph here, this optimal tension graph. It is only based upon two possible 3 layers of contraction. So you got isometric, which is contraction without movement of the skeleton, concentric, which is a shortening of the fibers, and then an eccentric, which is a lengthening of the fibers. And this graph on the left here only accounts for 2/3 of it and does not account for this history-dependent property. So yes, although you have less ability to develop tension concentrically and isometrically at a certain point, it doesn't take into account how muscles actually develop, act in actual movement. They use all 3 pillars with it. So this one on the right here, this is where Herzog and his team, they stretched a myofibril, which holds a bunch of sarcomeres, past overlap. This one down here is in the optimum. This one up here is starting to get leaned towards more of that descending limb, less optimum. And according to that theory, because of the like 100% of force differences between the two, the shorter sarcomere should pull this longer one past overlap, and you would see instability. But that's not the case. They're all parallel. So 2 big things here that it does not account for. So now Walter Herzog and his team, they have constructed a hypothesis for a new theory. This is the Titan filament theory of muscle contraction. So now this yellow piece here connecting myosin and actin, which previously has been shown to have passive contributions to muscle contraction, they're arguing that it has active contributions. So, and this is how. So basically, you got your actin filament up here, myosin filament up here, and this plus sign indicates the distal portion of Titan. And what happens is as you actively stretch that sarcomere, they have found that with the studies that they have conducted, that it shortens its spring length by attaching to actin. And you can see that from this graph here as it goes parallel as the sarcomere lengthens versus the origin actually continues to lengthen. And it's not known whether it just attaches or winds, but it definitely shortens, and it is doing something with actin. But these are the 2 theories proposed, either winding around or just attaching. So this means nothing if they can't prove that it does increase stiffness. Stiffness is the ability to have that residual force enhancement, where was it? Yeah, right here. So you have to have stiffness to have residual force enhancement, and you have to have stiffness to be able to have stability. So if they can't prove that this shortened spring length has stiffness, then it doesn't mean anything, but they were able to do that. So they did the same thing. They stretched sarcomeres past overlap here. So I'm going to go back to this graph. So overlap was at about 3.6 micrometers where the myosin and actin no longer interacts. Here, they stretched it past 4. And what they found was without actin or myosin overlap, and contrary to what the sliding filament theory would propose, instead of being equal passive and active traces, the active traces were 4 times greater than the passive traces, which makes this theory null and void. And the only way that you can produce tension without actin and myosin is this protein right here, Titan, which has been found to be one of the biggest proteins in the body. So I know I'm sort of zooming through this, but there's a lot to cover. I'm going to do the remainder of this presentation on the truths of all these components in the microbiological environment of Titan, and then apply this to ATG, basically. So truth is about Titan. There's 20 different proteins linked to Titan by diverse pathways. Way too much for me to cover. I'm only going to cover one. The MLP telethonin pathway involved in mechanical stretch and hypertrophy of the muscle fiber. Why is that important? This pathway has a nuclear translocation signal, and what is that? It basically means it's a key to the nucleus to create adaptive change in the environment. So it's very, very interesting to see that Titan has capabilities to be involved with certain pathways that can lead to adaptive change. Titan also has interactions with degradation systems of the cell. Why is that important? Degradation systems of the cell lead to turnover. Turnover refers to things that need to remodel their environment to adapt to a certain the signals that they're receiving. And so going back to that point, the N2A portion of Titan, which is the portion that is proposed to wind around actin in that theory, has been found to protect Titan from degradation. So interesting things about adaptive qualities of Titan. Going into like, how does that refer to like whole body things? You know what I mean? So like chronic training, how has that been shown to adapt? It's been shown to increase the ratio of Titan molecules from myosin, flanking myosin from 3 to 1 to 5 to 1. And what does that mean? So myosin right here, you only have 2 strings right here. So if in a three-dimensional view of it, you would have more strings coming out of there attaching to actin, which would translate into greater myofilament stiffness. And this stiffening could promote the effectiveness of accelerated contraction relaxation cycle. And what does that mean? How do you apply that? That just means walking, jumping, sprinting, things like that. You can improve that because you're able to handle more force. Tendon. So titin is in the muscle, which attaches to tendons. So clear evidence shows that muscle and tendon depend on each other to adapt correctly, and that muscle contraction shapes tenocytes. And that could be seen in this graph here. With normal muscle contraction, tenocytes, the elongations are longer and they're able to interact more with their extracellular environment. It's more organized. And on the right, without contraction, it's disorganized and there's less protrusions. Really cool to look at. Integrins, what are they? So these are proteins that sense tensile strain at the cell surface, and they're bidirectional signalers from the cytoskeleton to that connective tissue encapsulation, the extracellular matrix. And why are these important? They increase their concentration in response to muscle injury, and they're concentrated at the myotendinous junction. So another ability to adapt to forces at a location to where you can modulate tension. And I'll explain a little bit of about that later. Satellite cells, what are they? These are responsible for regenerating skeletal muscle after injury, and they are enveloped in that connective tissue encapsulation around the muscle fiber. And interestingly, they are connected right to the muscle fiber through the dystrophin complex. So another application of an extracellular component that can adapt and is connected to the muscle fiber. So laying out some final truths. Titin actually is able to also acutely adapt as well as chronically with that flanking quality that I've explained it to have. Integrin can stabilize the sarcolemmal membrane and prevent exercise-induced muscle damage with eccentrically trained rats. And satellite cells crosstalk with that connective tissue encapsulation, that ECM, to remodel its structure from the signals they receive from their environment. And one of the biggest signals they receive is tension. Tendons. So the tension that is developed when the cytoskeleton of tenocytes, which are tendon cells, can regulate the response to forces with gene expression. So they can change their genetic components from forces because of just their inherent qualities. And my favorite point right here, right underneath satellite cells, but it applies to all of them, this first point. It is known that different types of forces orchestrate the control of all biological functions. That is just crazy to me. Forces control all of these functions, including the ability of satellite cells to maintain cell and tissue homeostasis. So their ability to react to forces is in effect because of the forces is really cool. Summary on all these truths, getting to the nitty gritty. So the highest amount of force production, where does this occur? It has been found when titin is maximally challenged to remain stiff at long sarcomere lengths. Where does this force get sensed? Through integrins to the ECM and satellite cells, by titin to the nucleus of the cell, and by tendons through gap junction connections between tenocytes. What does the body love doing? Adapting specifically to the demands imposed upon it. That said principle in biological action has previously been shown in higher levels of exercise and things like that, but now this is being shown in the microbiological environment, which has just been amazing for me to just look into. What have all these components shown to do in response to tension demands? Increase levels of ability. Tendons with tenocytes, we saw it with the protrusions getting longer and interacting with more ECM. Titin with stiffness, we saw it both acutely and chronically. Integrin with stability, we saw it with its ability to resist turnover, and satellite cells with their ability to remodel that connective tissue encapsulation, that glue. So if anything else, this is the most important slide. If any of my rambling got lost, I'll try to make this clear. So my hypothesis on all of these truths, all of these studies were done with needle cantilever systems, which is basically they put there are two things there, they stretch it, they let it go back, and then they examine the results from it. Our joints don't work like that. Our joints are the needle cantilever system. They have three-dimensional movements. So my argument, this is where the argument comes in. I argue for challenging the ability as many titin filaments as possible to remain stiff through a joint-by-joint strength curve approach to strength training, which would elicit the most structural adaptation in all of these players to provide maximal resiliency for one's environment, from sports to activities of daily living. That's as simple as I think it can be. And I think this is where ATG fits in. So I didn't get to explain much about what their system is, but they use a bunch of different exercises that, according to the current state of strength and conditioning, they're like, oh, I wouldn't do that, I wouldn't do that, I wouldn't do that. So it's sort of on this, I don't know the word for it, but it's not normal in accordance to what is currently being done. But with the quadriceps musculature, this is just one muscle they use. They do it for all muscles. This is how they do it. So Here, the knee goes over the toe, the hip stays in one position, and the ankle stays in one position in the reverse step. And I believe that tighten filaments responsible for resisting this knee flexion and ankle dorsiflexion would be targeted here. But so this is still the same muscle. Now another exercise for it, to where now you're getting completely lengthening that quad over the toe with the hamstring covering the calf and the hip drop. So another angle to stretch that quad. And then finally the KLT squat, because the quadriceps muscle, I'm going to try to like do a little bit of an arrow here. So like it crosses the hip. So you need to extend the hip to get maximal stretch at that point. So that point you would be stretching tighten even more maximally, still getting that knee over toe. So all these knee over toe, knee over toe, knee over toe, but at different angles that challenge its ability to fight knee flexion. So call to action, where to go for here. I think future studies should be done to elucidate the exact signaling effect responsible for eccentric exercise. And my capstone has like the entire like the evidences of eccentric exercise moderately loaded being helpful for rehabilitation and training and things like that, but I didn't want to load it up too much. But possibly centered around this, the signaling effects of tighten because of its obvious involvement with stretch. And I think studies should be done with isolated, so muscle, tendon connections that determine different effects of stretching done in one direction, like those caliper systems or the whatever, the two things that hold the thing together and pull it in one direction versus multiple directions to see the beneficial effects and sort of like extrapolate that for the strength curve approach. And also, I think in a bigger sense, try this with tendinopathy patients with a common means of rehab, maybe an eccentrically focused rehab approach, and then a strength curve approach like ATG's methods to see if some sort of biopsy could possibly be done. Then you can really see the physiological mechanisms in that environment. So I know that was really, really fast. I hope you enjoyed it. That's basically it. The only source I really have is my honors capstone. It's linked here if you want to check it out. It's also on my Instagram and my bio. So I hope that was awesome for all you. I'm good for questions, I guess.